Okay, hello everyone. This concluding portion of the lecture on molecular biology and specifically transcription. We're going to try this out as we move some of our lectures out of the classroom and onto the computer so that you can watch them more than once and keep up this way. So what I'm going to do now is continue where we left off on Tuesday with the transcription lecture and the end of molecular biology. And then this will be up on YouTube. So what I'm going to do here is share my screen so you can look at the PowerPoint presentation. So now we're looking at a slide. So as you might remember, we were talking about mRNA processing in eukaryotes. And we talked about the three major processing events, which is the addition of the 5' cap, 7-methylguanosine, the removing of the introns, not the removing of the exons, known as splicing, and the polyadenylation of the 3' tail. And so we've talked about capping, we've talked about splice, we've talked about capping. Just to review here, the cap is this 7-methylguanosine. It's very much like the addition of an additional nucleotide at the 5' end of the RNA transcript. It's got these three phosphate groups here linked through phosphodiester bonds that link up with the first nucleotide through the 5' carbon. And so this 5' cap serves to protect the RNA from degradation and promote translation by the ribosome. So that's one major way that almost all messages are modified after transcription. And we're going to talk next about splicing, so the removal of the introns and linking up of the exons. And then as you'll see here, there's this poly-A tail. These poly-As are not encoded in the genome, and they're actually added on by poly-A polymerase at the end of transcription. So we stop there. We want to talk about splicing, and this was a very important discovery made primarily out of the lab of Phil Sharp at MIT, won a Nobel Prize, and so they were doing some work looking at hybridization of DNA and the RNA transcripts that are produced from those particular genes. And so this is an electron micrograph of a DNA RNA hybrid. And so what you can see here is that in certain regions, the DNA and the RNA are fully hybridized and fully complementary, and then there are sections of the DNA which loop out, which are shown in red, that are non-complementary. And it appears that those portions of the RNA, the precursor RNA transcript, were spliced out. And so you've got multiple loops here. I lost my pointer. This one here, this one here, this one here, that were all part of the primary transcript, but then were excised after RNA processing. And so oftentimes what happens in eukaryotes, especially higher eukaryotes, is that the primary transcript gets cleaved by the spliceosome, and the exons, the coding sequence is retained to make that mature transcript. So the spliceosome is the machinery